Well, good day, everyone. This is Dr. Rollin McCraty at the Institute of HeartMath, and I see a lot of people are still logging into today's webinar. So before we start the formal presentation, we'll give it a minute or two here to let those that are still logging in get logged in and synced up. It's my great honor today to introduce Dr. Carlo Ventura, who's become what I feel a very good personal friend and colleague, and we're starting to collaborate together to do, I think, some rather innovative research in the relatively near future. And I asked Carlo to do this webinar because I'm so fascinated by the, I think, really leading-edge research that Carlo's doing in the field of epigenetic information and how he's able to change the programming of stem cells through magnetic fields and sound vibrations. And he'll also talk about some coherent water work that he's doing. So Carlo, hello, and thank you so much for joining us today.
Thank you very much for the introduction. And first of all, I would like to thank you and the Institute of HeartMath for organizing this webinar and for the unique opportunity to be here with all of you. I would like to focus this presentation on several features of DNA, on the relevance of its architecture and shapes as driving forces that can modulate the expression of genes, unlocking the potential of any given cell to be transformed into the many different cell types that define the structure and function of our organs throughout the adult life. As you know, DNA is a very long molecule. It's about 2 meters long. And it is compacted in a very small nuclear volume with a diameter of only 50 to 70 microns usually. Only 1 or 2% of the overall DNA length contains coding sequences. That is, sequences that will give rise to a messenger RNA and to a protein that will in turn specify the structure and function of our cells. The huge remnant part was believed to be a sort of meaningless garbage structure until very recently. And it is now clear that cell commitment and differentiation is controlled by a complex interplay between cell signaling, the environment, and the continuous dynamic remodeling of this so-called garbage DNA into loops and domains. We are now understanding that we are facing an architectural DNA that is capable of acquiring a temporal and spatial organization, what we call epigenetics, shaping and specifying the multifaceted gene expression motifs that are part of the information of life. So the environment is important. We know now that even during pregnancy or childhood, we may have adverse events in the environment that can affect epigenetics, the nuclear volume and form and shape in the cells, as well as the transcription of our genes, ultimately leading to huge changing in epigenetics and in cell patterning and behavior. So the environment is important. And as you, Rollin, you're right. As you said, it's important to understand that when we say environment, we may also think about our mind, our consciousness, the possibility to rise our coherent state and change the environment and the way the environment may affect this epigenetics, the DNA structure and function. So within this context,
Carlo, if I may, before you move on, I think for some of our audience and listeners, it might be good just to talk a little bit about what the term epigenetics actually means. My simple understanding, and I want you to add to this and correct me if I'm wrong, because epi just means above or outside of the gene. Is that correct? So in other words, its influence is outside of the DNA itself in the genes that actually act as to program or control the gene. And this has been a revolutionary finding in that we're not locked in necessarily to our genetic code, that things in the external world.
It's a huge possibility of freedom that we have through epigenetics. So we're not stuck simply on the genetic code, but we are more and more free, and we can rely on epigenetics to change the function of our gene program. So, and within this context, stem cells may hold the promise for the rescue of severely damaged tissues that cannot be saved even by the most advanced pharmacological or surgical treatments. And this perspective has paved the way to a novel paradigm in the handling of complex diseases. Just think about cardiovascular diseases or neurodegenerative disorders. And this new paradigm is the so-called regenerative medicine that is using stem cells and cell therapy to rescue a damaged organ. However, as you can see in this slide, such hope mainly arises from the feature of totipotent embryonic or pluripotent embryonic stem cells that are able to self-renew and capable of producing all the progenitor cells that leads to many component cell types, including the end-terminally differentiated cells that make up the complete organism. So far, not only for ethical issues, but for immune rejection, tumorigenic problems associated with embryonic stem cells, most of the effort in regenerative medicine have been focused on the use of multipotent adult stem cells. Now, these cells exhibit a remarkably lower differentiating potential. They cannot be turned on in all the cell types of the human body. And their differentiation, their transformation is often not complete. So we're talking about cardiac-like cells, neural-like cells that are coming out of these multipotent stem cells.
So Carlo, if I may,
yeah.
Can we give, for me and I think some of our other listeners, it's just simple definition, or what's the difference between a totipotent and a pluripotent?
Well, a totipotent cell is a single cell, a fertilized egg, a fertilized oocyte that can give rise to the entire individual. A pluripotent cell is a cell like the so-called embryonic stem cells that can be transformed in any cell type that can give rise to many organs and tissues, but there is no possibility for a pluripotent cell to give rise as a single cell to an entire individual.
Okay.
A multipotent cell is somehow placed at a lower hierarchical scale level, as you see in the slide, since it can be oriented to become virtually any kind of cell. But usually this kind of transformation is not completed to become a real competent terminal differentiated cell. So the repairing potential due to these multipotent adult stem cells appears to be mainly related to their ability to secrete a number of molecules that turn on the inherent ability of tissue self-healing rather than, as I said, a true stem cell differentiation toward a given cell type. On the other hand, it is now increasingly becoming evident that even non-stem human adult somatic cells like hepatocytes or skin fibroblasts can be transformed or reprogrammed back to an embryonic-like state by viral vector delivery of a few 1 to 4 genes. So these reprogrammed cells that have been named induced pluripotent stem cells can be transformed into lineages in which these cells would never otherwise appear. And this kind of transformation is related to the acquirement of new conformation, new architectural domains in the DNA above the gene sequences. So once again, the possibility to reprogram even a non-stem adult cells backwards to a pluripotent embryonic-like stem cell is mainly associated with epigenetic changes.
Okay, so if I understand what you're saying, we could have a cell that's become, say, a skin cell or a fat cell or something like that. And the idea here in regenerative medicine is to take that cell backwards, so to speak, to a pluripotent state where it can then be reprogrammed to become a different type of cell. Is that what you're saying?
It's correct. It's like a time machine. You're reprogramming somehow backward this cell to an ancestral state in which any kind of decision is somehow possible. Even the decision to become virtually any kind of cell of the organism. And just think about the tremendous potential of this discovery. This is why the two guys that made this discovery, Shinya Yamanaka and John Gordon, got recently the Nobel Prize in Medicine. for their discovery that even non-stem adult cells can be epigenetically reprogrammed backward to a state where they can eventually give rise to neural cells, cardiac cells, skeletal muscle cells, or insulin-producing cells. So this is a really relevant step forward in science. Mm-hmm. But despite these new achievements, stem cell commitment and differentiation is still poorly understood, and it represents an extremely low-yield process, especially for a number of commitments that are long being considered as a major target in regenerative medicine, including, as I said, the cardiac differentiation, neurogenesis, or the transformation into vascular cells. Just consider that usually the differentiation of an embryonic stem cell into a cardiac cell is a very low-yield process. It's about 0.002% of the overall differentiating potential of the stem cells within the embryonic body. So the main message is that we need to talk to stem cells, or even to adult non-stem somatic cells, to reawake their healing potential. And so far, it was believed that only chemistry may be able to talk to our genes or to handle the epigenetic information stored within the DNA molecule. But I would like to tell you a different story. Not only molecules, as we say in this slide, but we can also turn on stem cell cardiac differentiation with physical energy. We first demonstrated that we can do this with extremely low frequency magnetic field. Here is the first page of the paper that we published in the Physical Journal in 2004 and 2005. And as you can see here, by exposing stem cells to a magnetic field, an extremely low frequency magnetic field with a 0.8 millitesla intensity, which is basically the magnetic field which is delivered throughout the power lines, we can trigger a number of genes that drive the cardiac cell commitment. That is genes that somehow tell to the stem cell, you're not going to be a non-differentiated stem cell anymore, but you will be oriented to become a cardiac cell. But more than this, exposure of our stem cells to these magnetic fields may also enhance the expression of genes that encode for cardiac-specific proteins. Proteins that, for instance, drives the contraction of the myocardial cell. And here you won't see the movie of these cells, but you can imagine on your left side that natural chemistry, like small peptides, may orchestrate the differentiation of stem cells toward beating cardiac cells. But we can actually do the same thing by exposing the cells to a magnetic wave, to a magnetic field. More than this, yeah.
No, go ahead. I mean, that's amazing that you're able to use magnetic fields to deliver, if I understand it right, information to the DNA that changes the chemistry, if I understand what you're saying.
Changes the chemistry, right. and it goes through the transcription of the genes. So we were able to influence also the expression of genes in nuclei that were taken out of the cells. So the magnetic field will act somehow by changing a sort of memory within the cells, and this memory was changed and was permanently changed even when nuclei were taken apart from cells. Later on, we moved a step forward and also using magnetic fields, but of different kind. We used the magnetic field of 2.4 gigahertz, like the Wi-Fi band, which is the same kind of magnetic field which is used in internet connection worldwide. And we have been using a device that we named radio-electric asymmetric conveyor, REAC, that was invented by Dr. Salvatore Rinaldi and Vania Fontani in Florence. And this device has the ability to modulate stem cell to differentiate or even transform an adult non-stem cell, like a human skin fibroblast, to behave like a stem cell. The physical underpinning of the REACT technology may be the following. Upon the interaction of two oscillators, namely two electromagnetic fields, a resultant oscillatory pattern may be generated. In actual fact, a resultant current and magnetic field. The exploitation of the REACT technology implies that among the interaction of these two oscillators, one is represented by the oscillating electromagnetic field generated by the cells or by the entire organism, and the other one by a weaker electromagnetic field produced by the REACT device itself. Upon the establishment of such a dual system, a resultant electromagnetic field or current is generated that induces the cells or the organism to respond in a sort of autologous fashion with cell-generated radioactive microcurrents that we think may ensue from an array of cellular ionic flux. So by the aid of REACT technologies, these autologous radioelectric currents can be focused and conveyed back to the area that we would like to treat in a patient, or to the cells that we would like to expose and eventually reprogram. So here is it.
Well, if I may, just on that, if so maybe help, because it took me a while to understand this as well. Because you just said a lot there that a lot of people probably wouldn't be able to follow. But if, as I more simply understand what you're doing there, is you're setting up a resonant loop or a resonant communication channel with the cell and the genes. Is that a way of thinking of what's happening here?
Absolutely correct. It's a kind of bioresonance. So we are picking up the endogenous signal that is generated by the cells or the entire body, and delivering backward to the cells or a tissue with a conveyor probe. This is mainly the basic underpinning behind the REACT technology. And what we did, we first started with mouse embryonic stem cells and exposing these stem cells just for 2 days to the radioelectric currents, and then culturing these cells for additional 7 days without any REACT treatment. And we were able to prove that the radio frequency energy loop was able to remarkably enhance cardiac, neuronal, and skeletal muscle differentiation, as shown here in mouse embryonic stem cells. So we started to provide evidence that radioelectric fields asymmetrically conveyed by this technology may represent a new tool for improving tissue regeneration. Actually, what we saw was the ability of this modulated magnetic field to prime the expression of genes that trigger and drive the differentiation of stem cells toward cardiac, neural, and skeletal muscle, shown here. And also specific cardiac, neural, and skeletal muscle proteins were upregulated by exposure to this REACT technology. And when we give a look by confocal microscopy at the intact cell level, we could detect the expression of specific neural marker protein or skeletal muscle microprotein, or specific cardiac-related marker proteins that specify the acquirement of a terminal differentiation state. And you can see here the huge difference in the number of spontaneously beating cardiac myocytes that we can achieve after treatment in the presence of REACT compared to the untreated control stem cells. As you can see here, the difference is very huge, not only in terms of the number of cells, but even the quality of contraction is completely different. Once again, I have no chance here to show the movie of these beating cells, but they do create a sort of pacemaker activity in the tissue culture, and also a specific wave loop form of contraction that very closely resembles what is going on in a real heart.
So in other words, there's cells that spontaneously start to beat in the culture and group together, kind of like what happens in a real heart. Is that?
It's a system that is quite good in terms of basic science, since it somehow recapitulates what's going on in the embryogenesis in any animal species, including humans. Then we moved on to human adult stem cells, since, as I said before, these cells are mainly actually used even in humans for regenerative medicine purposes, and so, as I said, these adult cells are mainly the basis for the future efforts in cell therapy. And we wanted to know whether the exposure of these cells to react-modulated magnetic fields may trigger the differentiation of these cells that, as I said, are not able to complete a program of cardiac, neural, skeletal, muscle differentiation. They only try to behave or become something close to a real cardiac cell, but they do not complete spontaneously this kind of trajectory. What we used a novel system. We started from fat tissue lipoaspirates that were processed in a non-enzymatic fashion throughout a novel technology that we named lipogems to harvest stem cells. And this technology was developed by Dr. Carlo Ventura, a plastic surgeon working in Italy and in the Diabetes Research Institute in Miami as well. And it's important that by using this technique, once again, we use mild mechanical forces to harvest the real microenvironment where the human adult stem cells live, which is a kind of particular vascular fraction where the cells stay within the vessel wall. And these cells are not stressed like usual methods that employ enzymes or collagenase to isolate these cells. So we took advantage of this kind of preparation that is called, as I said, lipogems. So this is the morphological.
If I may, just to clarify there, from what I've understood in talking with you, this is a revolutionary thing that you're talking about here.
Yes.
Because you're really able to collect stem cells from humans, their own stem cells for future treatment back in that person from their own fat tissues, if I understand that.
Not only collect stem cells, but even collect stem cells within their own real microenvironment. As you said, the environment is very important, and if you simply harvest stem cells by isolating these cells throughout enzymatic digestion, as is usually done, then you will have stem cells, but you will destroy their own microenvironment, and you will somehow unlock their cell signaling network from their epigenetics. We know now that harvesting stem cells within their own environment is equal to make a revolutionary strategy of tissue transplantation. The stem cell microenvironment is like a tissue within the tissue, and it contains the stem cells. But as you can see in this slide, by simply placing 1 milliliter of this microenvironment in a tissue culture medium, you can release stem cells that can attach to the tissue culture dish and then start to proliferate up to confluently, as shown here. So we decided to use this kind of approach because the lipogems approach is, to our knowledge, the best way to deal with intact human stem cells. And even in these circumstances, the exposure of human adult stem cells to react-modulated magnetic field was able to trigger the expression of a cardiogenic gene program, as shown here, or the expression of genes that trigger the differentiation toward a neural fate, or the differentiation into skeletal muscle cell. In other words, we were able to turn human adult stem cells into embryonic-like stem cells that are able to express a panel of genes for this kind of complex differentiating patterning.
So
and once again,
Carlo, I'm sorry to interrupt you, but this is what you were talking about earlier, where you're able to take a cell that's become some type and take it backwards to more of a stem cell so it can be reprogrammed into the type of a cell you want it to become. Is that correct?
Yeah, it's correct. We're still within the frame or the context of the stem cells. So we are reprogramming a multipotent stem cell backward to a pluripotent embryonic-like stem cell starting from a human adult cell, so a human adult stem cell. So this is important because we can, as shown here, transform these adult human stem cells into cardiac cells, as shown here, or skeletal muscle cells, or neural cell. And this was achieved by exposing a human adult stem cell to radioelectric fields delivered by the REACT technology. And we moved a step forward, more forward. As I said before, the Nobel Prize in Physiology and Medicine has been recently attributed to these investigators, Dr. Yamanaka and Dr. Garden, that discovered that by delivering a few genes, 3 to 4 genes by viral vectors into human adult fibroblasts, which are not stem cells, these are adult somatic cells, these cells could be transformed backward into an embryonic-like state. So they were speaking about induction of pluripotent stem cells, as you see here. So this was a revolutionary discovery, not only because this says, once again, that the environment is so important, because these genes that were introduced into the adult somatic cells were encoding for proteins whose role was mainly to change the shape of DNA. So by changing the shape of DNA, by changing the epigenetics, once again, you can set the clock backward in an adult somatic cells to become an embryonic-like cells. So the idea was also to work with fibroblasts and with cells that are located within the scar tissue to eventually use the scarring tissue to rescue a damaged organ, which is a totally revolutionary idea. Unfortunately, this kind of approach was afforded by the use of viral vectors that were modified HIV. So their use is not readily envisionable in humans due to the risky and the side effects. Moreover, these cells these reprogramming cells were somehow frozen into an embryonic-like state with a very, very low yield of subsequent redifferentiation into, let's say, cardiac, neural, or any other important subtype. So the contribution to the issue of regenerative medicine or future cell therapy approaches in humans was just an expectation, a big expectation at the beginning. What we did is to expose human adult fibroblasts taken from human skin to the radioelectric fields conveyed by the REACT technology. And we were able to show that these cells were directly transformed into cardiac, neural, or skeletal muscle cells, simply with physical energy, without the use of viral vector or expensive synthetic chemistry. This slide just shows you, to cut a long story shorter, that the exposure of these cells, these human adult somatic non-stem cells, to the REACT field was not affecting the cell viability or the survival of these cells. But the exposure to the radioelectric fields was able to once again, drive the expression of genes that commit these cells to a cardiac lineage, or drive the expression of genes that specify the differentiation into cardiac myocytes, or
if I may,
into, yeah.
Well, I'm going to back up just one thing that you were saying there, because I think it's an important thing, and I didn't really understand this until we had more chance to talk, you know, in private at your lab when I was visiting you. But the significance here, as I understood it, Carlo, is that, like, say, in somebody who's had a heart attack and they have a lot of scar tissue on the heart,
yeah, absolutely,
that you can turn that scar tissue back into a functional heart muscle. Am I saying that correctly?
Correct.
So
not only heart muscle, but as you can see here, we were able to drive the expression of neural genes or skeletal muscle genes, and any damaged organ has a scar. So the brain has a scar, the skeletal muscle has a scar, the pancreas has a scar, and diabetic subjects, and so on and so forth. So the idea is to consider the scar tissue as a kind of aberrant attempt of the organism to repair, to rescue, but without completion. And by this kind of exposure to modulated magnetic field, we may eventually make this kind of effort, natural endogenous effort, complete to the rescue. Importantly, this kind of modulated magnetic field delivered through the REACT technology, we're able to time-dependently regulate the expression of genes involved in the reprogramming of these skin fibroblast cells into pluripotent cells. All the genes that you see here were the genes that Yamanaka was able to induce in human skin fibroblasts with viral vectors. We were able to do this simply with physical energy without any risk related to the use of viral vectors.
Right,
More
by
than
physical,
this,
but just slow down for a second here. And by physical energy, you're really meaning the magnetic fields.
The magnetic field, absolutely the magnetic field. And more than this, as you see in the slide, these genes were first upregulated on the right side of the graph, and then downregulated. This is very important because we now know that in order to achieve a high yield of subsequent differentiation of these reprogrammed cells, the same genes that were triggered to achieve this kind of reprogramming must be shut down in order to allow an efficacious terminal differentiation. And as you can see here, we were able to obtain a huge amount of cardiac cells, skeletal muscle cells, and neural cells, starting from a non-stem human adult cell that was exposed to the modulated magnetic field. And it's very important because in the Yamanaka experiments, the final yield of this commitment is below 1%, even lower. Here, as you can see, we get a huge amount of terminally differentiated cells, usually between 15 or 20% for each lineage. That means that we only left maybe 20% of non-differentiated cells. Now, another important point is that the non-differentiated cells that we can live with this kind of technique were simply human fibroblasts. No embryonic stem cell anymore. This is important because otherwise having a huge amount of non-differentiated embryonic-like cells is a tumorigenic risk. So once again, this kind of strategy using a magnetic field to talk to the stem cell epigenetic is more safe than the usual way of delivering a gene by a viral vector or using a chemistry to modulate the gene expression, because in these last cases, you're not avoiding a tumorigenic risk. I don't know if I make this point clear. But there is another possibility to reprogram a cell with physical energy, not only by using magnetic field, but even with sound vibration. Before going further in this discussion, I also would like to remind that our results are also in keeping with the use of radio frequency waves with the one that we applied that correspond to kind of harmonic of a therapeutic method that Dieter Breuer has developed within two universities in Berlin, in Germany. And we also intend to cooperate within mutual projects in the future. Dieter Breuer is currently investigating also the relationship between the cosmic and the terrestrial electromagnetic fields and their effects on the state of mood and consciousness of man. And this is also what it turns out in his film Solar Revolution and in his book. So once again, we're talking about a very huge environment, which is the cosmic or the terrestrial electromagnetic field that can conceivably affect our behavior, our mind, our consciousness, and as a consequence of that, it may also interact with epigenetics and DNA. And this is once again what we would like to explore together with you, Rollin, and the Institute of HeartMath. Now, another issue that makes the role of the physical energy and the environment even more consistent in terms of the potential of affecting cell reprogramming and epigenetic is the vibration, the acoustic vibration, the sound vibration. Here, once again, I don't have a movie running, but if we look at cells and their inner structure, what we call the cyto and nucleoskeleton, which is made by fibers and tubi, we will never see something static, but something really vibrating. It's a continuous vibration with specific frequencies and harmonics. And also within the nucleus, you can see here, within the nucleus of cells, you can see here with this kind of scaffold, which is not static, it's continuously vibrating and remodeling. And you have to imagine that the DNA itself is rounded up within this scaffold. So all the world of cells is vibrating, and it is vibrating in resonance with the extracellular matrix as well. So it's a world of continuous vibration. And this kind of vibration can also be perceived by specific equipments like the atomic force microscopy, AFM, which can also detect the nanomechanical vibrational patterns that is arising from cells. And by working with a dear friend of mine, James Ginczewski, worldwide renowned physicist at the University of California, Los Angeles, we are now collaborating on the issue that there may be a sort of nanomechanical signature during stem cell development and patterning. The work that Ginczewski and we have done also provides evidence that cells can emit specific vibrational patterns that can be recorded by the atomic force microscopy as sounds. Here is an example of the sound that is emitted at different temperatures by a single cell. So you can actually, I hope you can hear the sound of a single cell at 22 centigrades. Or sound, sorry, hope can. No, it's not working, unfortunately.
Well, we heard the first one okay.
Yeah, I hope this one, let me check again.
Yeah.
Okay, this is 26, 30 centigrade. And if you simply kill the cell with the tip of the microscope, it's just a sound. It's not an harmonic vibration anymore.
It's just static.
So our idea was just to record the sound that is emitted from a stem cell that is undergoing a specific differentiation, let's say cardiac differentiation, and see whether this sound may be somehow different from a sound that is emitted from a cell that is taking another route, like neural or skeletal muscle or whatever. And our first line of evidence is positive. Of course, this is a kind of work which is still going on, and we are currently investigating the effect of the sound of the heart that was recorded by a worldwide famous jazz musician, Milford Grace, who lives in New York, and apply this kind of sound to stem cells to see whether we may eventually turn on specific route of commitments from these cells. So it's a totally new and area of inquiry, the idea of using the nanomechanical vibration to drive a specific differentiation or cell reprogramming. But once again, it tells us about the importance of the epigenetics and the shape of DNA.
Before you go on, if I may, I just want to kind of slow frame a little bit and let people digest what you just showed this and actually played, because we always hear about, you know, how the cells are vibrating and this and that. But what you just did is actually literally recorded the vibrations of cells using very advanced technology, and we just heard the sounds that cells make and how the pitch of the frequency of the cell, the vibration of the cell, is changing as you change the environment around the cell. If I understand this right, is that correct?
It's correct. It's correct.
This is amazing. I just wanted for people to really understand what you just told us, and this is probably the first time anybody's ever actually heard a recorded sound that the cells are actually vibrating at.
Yeah, and also you have to imagine that the so-called cardiogenesis, the primordia of the cardiac differentiation, is the first morphogenetic event occurring in any species, rodents, amphibians, primates, and human as well. And it's amazing that the heart starts to vibrate. The primordia of the heart starts to beat, so it must emit somehow a sound. And also, it's intriguing that due to the spontaneous beating, there must be some radioelectric current and magnetic field as well. So once again, a sound and the magnetic field, a vibration and the magnetic field, or an electric current may work together to establish what we call information or a morphogenetic field, which is driving the fate of our organs. And once again, it's so important, the work that you're doing on the coherent state, on the HeartMath variability, and the possibility to enhance the consciousness and the coherent state, because once again, this is not simply environment in generic terms, but it might be the environment that talks to our DNA, and it changes the features of DNA, and eventually may give us a further chance to use practice to achieve a coherent state, to also achieve maybe in the future self-healing processes in our body. We will see. This is, I think, very, very important for the future. And here is a direct image of what the DNA looks like when it is visualized by atomic force microscopy. You see this little dot spot here indicated by arrow. This is a transcription factor. A transcription factor is a protein that somehow changes the conformation of DNA. And it is crucial for driving the function, what we call the expression of our genes. Now, as you can see here, every time a transcription factor interacts with the DNA, it creates this kind of loops. And we can measure the strength of this loop, the vibration of this loop as shown here. So once again, these loops or domains are not static. They are dynamically remodeling themselves. They are vibrating, so they can emit eventually a sound which is so subtle that we cannot detect so far. But what's important to stress here is that what we call a gene may be just this tiny part of this structure. The rest is just architecture, it's just epigenetics. And all these loops are epigenetics. These loops do not contain any coding sequence, any gene. The gene is just a small, minimal part of the structure. So it works just because of this shape, because of this dynamic architecture. And this is the environment of the gene. This is above the gene, as I said. And this is epigenetics. And we can talk to this epigenetics by magnetic field, by energy, and by changing the environment. Keep this. Another important step is the fact that water is maybe really crucial for our internal dynamics. We have to imagine that 77% of our weight is made by water, and if we count the number of molecules, water is about 98 to 99% of all the molecules in the body, and so it has a crucial role in cells. And it's also important to stress that when we talk about water within cells, this water is not water like the bulk water, but it's a water somehow bound to a surface. And we have many surfaces within the cells where the water molecules can bind to. Just think about the cellular membranes, the sarcoplasmic reticulum, the nuclear envelope, the cytoskeleton, the microtubuli, the DNA itself, all the proteins that make a contraction. These are all hydrated molecules. So they are tightly bound with water. And the water that is bound to these molecules, it can be considered as coherent water. What does that mean? When we say water, we can imagine a short range radiative band, which is the H2O molecule, as we all know. But we can also figure out that an ensemble of water molecules can somehow pulse and dance at a unison within a given environment where these molecules are bound. A protein of the cell, a tubulus or scaffold within the cell. So we talk about coherent water because this water somehow can be visualized as a long-range radiative ensemble of molecules. Now, a fascinating breakthrough was provided by the group of Emilio Del Giudice and Alberto Tedeschi that were talking about this coherent water form since many years. And later on, the Nobel Prize Luc Montagnier came up with this paper in the Journal of Physics talking about DNA waves and water. So basically, it's like saying that water can be signalized non-locally with kind of biological information to generate coherent domains capable of transferring informations. So what they did is a simple crucial experiment. They took a tube containing a DNA molecule from a bacterium. They put this tube into a coil, so they exposed the DNA within the tube to the magnetic field that was close to the Schumann resonance of 7.83 hertz, and upon specifying the DNA dilution, when it was very diluted, they could observe the emission of an electromagnetic signal from the water containing this diluted DNA. What was totally amazing is that when they put another tube above to the previous one, just containing water and nothing else, also the second tube started to emit an electromagnetic signal after 18 to 24 hours of exposure. And when they put all the building blocks that are needed to create a new DNA strand molecule, they were able to come up with the same original DNA sequences. So that means that somehow water nanostructures and their electromagnetic resonance can faithfully maintain and propagate DNA information. And once again, here a major role of a magnetic field is highlighted, because if the system is somehow shielded by a foil moon metal, the experiment cannot be reproduced anymore. So basically, I hope to have given you the idea that the cell can be a very dynamic and flexible entity. That the DNA is important, but the information is not simply stored within our genes, but it is stored also in the shape and the architecture, and that such architecture somehow can talk with the environment. It is part of the environment, and the signaling may also be featured in a kind of non-local fashion. And this is, I think, the last frontier to be developed and to work with, which is trying to understand this new paradigm of quantum non-locality signaling. And I would like to conclude my presentation with a few sentences that I was sharing with James Inceski. It says that in both the philosophical and the visual sense, seeing is believing does not apply to nanotechnology. We may say to any extreme analysis within quantum physics or molecular biology, for there is nothing even remotely visible. And on the atomic and molecular scale, when we go deeper and deeper, data are recorded by sensing and probing in a very abstract manner, which requires complex and approximate interpretation. And more than in any other science, the fantasy, the creativity, the curiosity, and the ability to come up with a story of information becomes necessary to describe what is sensed and not seen. And once again, probing and sensing may be a new route throughout which we may enhance our coherent state and change our environment and our DNA. So information is once again the essence of life, and it's part of the DNA code in the sense that we have once again to consider the epigenetic and the environment. And based on this, also our institute, the Visual Institute of Developmental Sciences, that I have the honor to chair in Italy, is based on this view. Vid comes from the Sanskrit vida, that means perceiving things with different eyes. And it's also the Visual Institute of Developmental Sciences. It's our effort to work within the field of continuous remodeling and transformation, hoping that the science is not going to be an envelope anymore, but it's going to be something developmentally connected with our consciousness and with our heart. And I would like to thank you and thank everybody for joining us, and all the people that have contributed to this data. First of all, I would like to thank once again Rollin McCraty and the Institute of HeartMath. I would like to thank all my co-workers at the National Institute of Biostructures and Biosystems, the VID Laboratory of Molecular Biology and Stem Cell Engineering, the Rinaldi Fontani Institute, Salvatore Rinaldi and Vania Fontani, who were the inventor of REACT technology. People work in the Department of the Biomedical Science at the University of Sassari, the quantum physicists that helped us in formulating all the hypothesis and interpretation of data, especially in the context of quantum no-locality signaling, the Department of Chemistry and Biochemistry at the University of California, Los Angeles, Dr. James Zinseski. And I would like to thank Dr. Abdullah Abdulgader, the Prince Salman Cardiac Center in Saudi Arabia. He is doing fantastic work in collaboration with Rollin McCraty just on the effects of cosmic and terrestrial electromagnetic field. And this is going to be for sure a wonderful opportunity for future collaboration. And the Cell Transplant Center and the Diabetes Research Institute and the Worldwide DRI Federation, the Middle School of Medicine in Miami, Camillo Ricordi, and Carlo Tremolada, that I would like to remind is the inventor of the Lipogens device that provided us with so nice human adult stem cells. And once again, thank you for joining us.
So, Carlo, what a wonderful presentation.
Thank you.
Maybe take a couple of minutes here,
Sure.
because I think part of what I know you and I have talked about in our collaboration, just to help give people a perspective here, I think it's been absolutely proven now that epigenetics is very real in many labs, not just yours. And some of the, I think, very important findings, we were talking about this before the webinar started, for example, that's been done at UCLA, a number of speakers, Stephen Cole and people like this, that have shown that in early childhood, if we're in an adverse environment, a lot of stress and things like this, that leads to epigenetic changes that are with that, as the child grows, that are with them for a lifetime. And that basically those type of changes are down-regulating all the processes in our body that underlie our ability to have strong immune systems, and increasing the processes that create inflammation. So this is finally given the link, the understanding to why early childhood trauma, for example, can cause many, many problems later in life that we're even starting to see in heart rate variability recordings and things like this that show up in specific ways. So that's, I think, pretty well established now for those that really understand where the state of the art is. But what we're suggesting here is that it's not just bad things. That
we can do the reverse.
We can do the reverse, exactly. And as we become more coherent in our language,
absolutely,
we're changing the energetic information in our system. This is what we hope to prove in our collaborations, that also leads to epigenetic changes that sort of open the DNA to express new things. And
I'm sure that this will happen. I'm sure, because in many issues we're facing a reverse remodeling. So the story can go both ways, and we can do the good things and the reverse of the effects of the bad environment. We can create a new environment, a positive environment, an environment where we can increase our consciousness, and people can practice to achieve this. And I think our goal is going to be just to prove that we can go down and down up to the molecular and quantum signaling level.
Yeah, absolutely. Could you go back a couple of slides in your presentation? There's one other thing I'd like to just click on a little bit, right there. So this is a rather profound experiment that you described. I know you went through it quickly, but I think in the original papers they even used the word teletransportation of DNA, if I remember correctly. But this, this was profound with what you said, where they had DNA in one test tube and just later a test tube with just water in it started vibrating with the same signature of the first test tube, and then when they added the components of DNA, not DNA in any way, shape, or form, just the molecular substrates that DNA formed that almost exactly duplicated the DNA in the first test tube. Is that correct?
That's correct. That's correct.
That's rather profound when you think about it.
Yeah.
And what I think it's illustrating, at least from one perspective, that that ties into some of our other work with the Global Coherence Initiative is that the magnetic fields, and it's interesting they used exactly the same frequency of magnetic field that the Earth is vibrating at. As the carrier of that biologically relevant information that caused that teletransportation. Am I making sense?
I absolutely agree. Yeah, it makes a lot of sense.
So, kind of an abstract way, it adds support to what we're saying on the Global Coherence Initiative work, that the Earth's field acts as a carrier wave that connects us all.
Yeah.
Right?
And also, it raises the issue of non-local signaling, which is also very important.
Exactly. So I think this is relevant, and you also mentioned earlier Dieter Bohr's work, who we're also collaborating with, and this is a subject very close to his heart, of course, who believes that the Earth's energetic environment that we were talking about briefly here is important in modern days of helping expand consciousness, with the changes that are going on. And I just wanted to mention that Dieter does have a wonderful DVD, a movie that he made called Solar Revolution, which you also mentioned.
Yeah.
But a lot of we have a relationship with Dieter where we're actually giving those DVDs away for anybody who actually wants to help donate or fund to the Institute of HeartMath, can get a free DVD to help support the very kind of work we're talking about here in this kind of research. So it's a current fundraising campaign that I know our institute is doing. So it all ties together.
Wonderful.
Points I wanted to make here. So any last comments you want to make? Parting comments?
No. I'm fine. Thank you.
Yeah, well, thank you so much. I know it's late in the evening for you over in Italy, and very much appreciate your taking the time out of your very busy day to an evening from your wife, who I know you're getting ready to have another child in the near future.
Yeah.
To do this with us. And I did have the pleasure, and it truly was a pleasure, to spend a couple of days with Carlo at his lab. Absolutely fascinating lab. I was really surprised to go in and realize that your lab is in a building built in the 1400s, was it? With all the paintings on the ceiling and the walls that date back to the 1400s. It's just really a very neat environment that you have to work in. Well, thanks again, Carlo, and very much appreciate it.
Thank you very much. Thanks a lot.
You have a great evening.
Bye-bye. You too. Bye-bye.